Do you guys have a tutorial on your course materials called Peptide Atlas tutorial? Okay, I think it'll help a lot to go along. So as you saw last time with any web-based tutorial, there's often things, or any live tutorial, there's things that can go wrong. And sometimes it's the software, and sometimes it's what you put into it. So if you could all navigate to the homepage of Peptide Atlas. I talked about Peptide Atlas the other day. It's actually a resource that's been around for a while. It has a number of interesting features. It's probably most amenable or most useful to people who have some organism or some set of proteins in mind that they want to explore. But there are a lot of, if you have a question to ask, there are a lot of resources available. So on the homepage, you can see there are quite a number of links. The main links are across the front of the page. These are some of the related resources. The SRM Atlas, as Ulrika just went over, and the SWAT Atlas that Sam will go over later, as well as the PASL SRM library. I'm afraid that the very first link that I'm going to ask you to open after this is the repository link, and I happen to know that it's actually quite slow. So go ahead and do what it says in number 2 here, which is click on the data repository. So that link is on the left navigation bar right underneath Atlas data. And so what this is doing is it's going off and fetching a number of experiments. And it turns out that it's a sufficient amount of data that it, over the wireless network, at least yesterday, it was taking quite a while. So I suspect you may have some difficulty loading it. But this is what it looks like as we went over before. So what's easier to see, the mouse or the pointer? Doesn't really matter. So each accession has this PAE accession, and I'm going to go ahead and open up this one. And you can see that underneath that experiment there's quite a bit of additional information. So has anyone successfully loaded the page? No? Still loading. So probably after about a minute, it's going to time out and tell you that there's no luck. But if probably the first couple of you that got in may have some success. So as it turns out, what this is doing, it's actually going and fetching the entire list of experiments each time. So we can actually limit the amount of data that we're asking for. So does everybody have sort of the upper part of the page loaded where it says search all and there's like a little tab? That's yes or no? Okay. So we can actually enter some text in there. So this one suggests that we enter in, we type in yeast into the search box. We could actually type something a little bit smaller. So you see the first one up here, this raft APR. Why don't you try typing that into your search box? RAFT APR, and then search. And since this is a smaller subset, I'm hoping, okay, well, apparently I've misspelled it. Oh. How do I do that? Control plus, sorry. Okay. So when I do that, it actually came back and it brought me back just one experiment. So can you guys try to do that? Type in RAFT APR and hit enter. And it'll come back. Was anyone able to see that? Okay, very good. So what you'll see is once you've done that, you can open up the section, and there's some information about publications and whatnot. And at the bottom, there is a link to download, but I can't quite get there. So let's try a search that has a little bit of something else. And you guys can type whatever you want. If you have MTB or something that's of interest to you, why don't you just type it and see. I'm going to try jerkcat. And the reason that this is working a little bit better than the previous one is that I'm only asking for a subset of the data. And it turns out that this is making a round trip to the server each time. So if anyone has actually made a successful search, please raise your hand. Okay, so this is what you would do if you were interested in one of the raw data sets that is up here. Yeah, so down below it's the individual experiments. So if you'll go back to the main page of Peptide Atlas and go down the page, I'm gonna make this a little smaller. Can you guys still read that or not? Because I can't. The mouse, I'm not used to this mouse. So basically, if you just put your cursor into this search box and hit enter, it will take you to the main search page. And once you've done this, you see this set of tabs up along the top. And I'll go ahead and make that a little bit bigger. So basically, this is a menu that allows you to get into the atlas. This is where all the different functionality resides. And as Ulrika showed you, underneath the SRM Atlas tab, there's a number of things related to SRM. So the first thing we're going to do is click on the All Builds tab. And if you click on the Select Build link, it will load up a list of all the builds. And it turns out that this isn't even all of them. If you click on this little plus, you'll see some additional builds show up. But there's already quite a few to begin with. So what we're going to do is take a look, once this is loaded, take a look down the page and go down to the Yeast 2013-03 build. And one thing you'll notice is that as you scroll down, one of your builds is automatically selected. And for me, it's the human, and that's going to be your default build. So anytime you're doing something in the Peptide Atlas, it defaults to a particular set of data, so a particular build. And so sometimes if things aren't working, you will need to change that. But most pages where you would actually want to, you will be able to make that selection. This is a place where we can see an overview. So go ahead and click on, if you were to touch this radio button, then anything subsequently you would do, that would be your default build. And it says that it's been selected to Yeast 2013. Whereas if you click on this link, it's going to take you to a page of some information about the build. And basically this is taking and sort of summarizing across all the different experiments that are in the build. And it's a little bit slow, sorry. So at the top section you see information about the build itself. It's from yeast, the number of experiments that it was from, the PSM-FDR threshold that you guys are now much more familiar with, the number of peptides and the number of proteins. And again, this is a very precise, conservative estimate of the number of proteins, excuse me. As Ulrika showed you yesterday, there's often a link to show the row descriptions, and you can see if you have any questions about what those are. And in addition, you can also toggle shut these sections. And sometimes if you're looking for something in particular, it's helpful to toggle those shut so that you can sort of go straight down the page to what you're looking for. I'll draw your attention down here. And again, this is a plot that shows all the stuff, all the experiments that went into the build. So can anyone tell me why this particular experiment is especially wide? Do you remember what's on this axis? Yeah, so it's the number of spectra, exactly. So the top of this at any one point is the cumulative number of peptides there are, and the blue is the number of peptides in this particular experiment. So this experiment that's narrower and has a higher blue bar than this one is actually, in a sense, well, it's a more productive. A higher number of the spectra received a match over threshold, so it's potentially a better data set. And as I showed you also, down at the bottom there's a heat map that shows the different peptide accessions across the different experiments. And one thing that you can see here, you see this streak of really dark information. So this is from the Matthias Munn yeast data set, which is very large, and so it turns out that that was a big contributor to the atlas. Okay, so I'm going to go back to the top. So I wish I could show you this. So I found the tutorial itself a little bit dense. So I broke it into sections, but I can't display that. For some reason, it didn't want to read my Macintosh file on here. But basically what we're going to do is we're going to go to search database on the left side. And we can also do that by clicking on the search link at the very top of the page. So once you're in this, so Peptide Atlas is a resource, links out to a lot of different places, and I admit that it gets quite busy at times. So once you're within Peptide Atlas and you're looking for functionality, almost everything is actually available through these particular links. So if you click on the search, it's going to bring up this search box. And so right now, it's going to search in any type of build. You can restrict it to whatever species you'd like. But it is worth noting that each organism has its own accession space. And so if you don't utilize an accession that makes sense to that organism, you're not going to get any results. If you're in doubt, then typing in any is a good way to make sure that you see something. So the instructions are to type in YDR502C into the search box. And we'll just leave the build type as any, and we'll click Go. Now that happens to be an SGD accession, and so that is very specific to yeast. And so almost certainly what we're going to get is, yeast matches. And you see here that we have different resources. Up here it says exact search result. If I were to enter a smaller, a more ambiguous string, say a gene name that's found across many organisms, what you would find is you would find results from different organisms, but you'd also often find that there are wildcard results. And this is basically using whatever you typed in as a root and then adding wildcard characters and doing a more expansive search. And that happens by default. So I want you to go ahead and click on the main atlas. So this, if you click on the identifier link here, you're going to go off to the page. And sorry if you're way ahead of me, and basically, this is what I showed you before. Again, there's a lot of information here. Protein name. There's a protein group. There's a set of orthologs here that we can go off and explore. And so this is a set of orthologous proteins across various organisms. And you can see that we have for C. elegans, gosh, what the heck is that? So for human, mouse, yeast, and C. elegans, we have these. And at the bottom, there's a clustral alignment that shows the similarity. And actually, this is a very highly conserved protein. So you can imagine over multiple organisms, it's quite common to have conservation, but that's not quite as significant at the amino acid level. But if you look at the bottom row here, this is the consensus. And the way clustral works is if there's an exact match across all of your sequences, you get an asterisk, a star. If there's a conserved match, which means the amino acids are functionally the same or similar enough that they're considered in the same class, you get a dot. And so this region right here is very highly conserved. The N-terminus, not as much, but right in through here, it's very highly conserved. So I'm going to go back. So looking at this page, can you tell me what the coverage is of this protein? And I'll give you a hint. It should be down. It's down here. So we actually have several different representations of the sequence. Again, this is the different peptides laid out on a track. And the way this works is if you have multiple sequences that overlap, it basically knocks one down so that you get a broader plot. So if you just had a few peptides across this whole thing, it would be much narrower here. But this is a way to represent within the context of the sequence space which peptides we have. So then there's also this color-coded coverage map, and you can see that almost all of the amino acids in this peptide have been seen by at least one, in this protein have been seen by at least one peptide. So can anyone see where it says what the coverage is of this protein? Yes, so it's 93.7%, and then there's another number here. So it's down at the bottom of this coverage plot, and it says 93.7% and 98.3 of the likely sequence. And what that means is these peptides that are considered unlikely are simply too short, or in some cases too long, to have been observed. And so really we have effectively 100% sequence coverage, or very close to it. So underneath the set of peptides, so this peptide view shows you the peptides. Yes? Absolutely, absolutely. Yes. So actually when Peptide Atlas was first built, it was very trypsin-centric. But we have changed the designation to be number of enzymatic termini. In most of our builds are purely tryptic, but we do have some builds that are mixed, and in that case we adjust this percentage. So this particular set shows all peptides arrayed in the context of the sequence. The ones that are more brightly colored have been seen more often. And down here you see basically the ranking of the peptides, so the peptides that have been seen the most. And right now they're sorted by this suitability score, which is a measure of how good basically how many times the peptide has been seen and how many samples it's been seen in. So how representative of this protein across all our experiments is this particular peptide. So the top-ranked peptide by number of observations, can anyone tell me what that is? Right, so in order to do that, you actually have to sort by this. Because if you'll see, there's one down here that's about 1,300, but if you sort by number of observations, so by suitability, the ESS, it is lower down. But by number of observations, it's only second. But you're right, the first one is this 1471. So there's a little bit of text here that says MGL, and there's a definition of it at the bottom. But basically that means multiple genome locations. And the reason that that was less highly rated is that it is seen in other proteins, and so it's not purely indicative of this particular protein. And so we essentially, Peptide Atlas is very peptide-centric, and so we essentially demote it because it has this multi-genome locations. Another characteristic of a peptide that would get it demoted is MC, which is missed cleavage, and NTT, which is really number of enzymatic termini. NTT actually stands for number of tryptic termini. And again, ideally you will have 2 enzymatic termini. Even if you're on the N terminus or the C terminus, you will have that counts as one of your tryptic or enzymatic termini. The idea being is that we want to have peptides that people will see reproducibly if they do the experiment again. So if you take a look at these peptides, you see that there's this funny-looking PAP number, and that's the peptide accession. So if you click on the PAP 18396, 18214. So it's in my list, it's a little ways down the list. Can everyone find this particular peptide, this PAP 18124? You can also search in the page. So did you guys see what I did? I sorted by the number of observations. So these funny little triangles, as you've probably seen on other web pages, are a sorting mechanism. So I could sort alphabetically by peptide by clicking on the sequence. And that's going to give me the sequence sorted, descending, and unfortunately it doesn't take me right back down to the bottom of the page. So when you do that down arrow, that's a descending sort. Yes, sir. Yes, ma'am. Pardon me? It's giving an error when it's sorting? So you clicked on the triangle? Sorry to hear that. So So it's possible, so that actually does do a page refresh, so it's possible that there's some latency getting back. I am able to sort right now. So I'm going to sort again by number of observations. So if I sorted this way, I would be from the smallest number to the largest number. If I sort this way, I'll be from the largest number to the smallest number, and the triangle depicts that. Actually, we're not in the yeast triple queued. Just the regular yeast, I think. Oh, no. Yep, so I'm sorry. I'm in the wrong atlas. I apologize. You are correct. Actually, so I'm going to show you something. So it turns out I selected the wrong atlas. I'm going to go down to here, and I'm going to select the triple quad atlas. So as I said, whenever you do a query for a protein or some other entity within the peptide atlas, there's an implicit build. And in the pages where it's useful to be able to do so, we actually have a way to change that. So I'm just going to change that. And as soon as I change it, it's actually going to go back and it's going to try to reload my page. And so again, this is a much more tractable number of peptides. You see there's only probably a dozen or so. And again, the ones with the most observations are sorted near the bottom. So if we sort by number of observations descending, that's the downward facing triangle. You'll see that the top hit is this FVIGG. Is that what everyone's seeing? Okay, anyway, let's go ahead and click on that particular one, the 0048180. Do people see that one? So I'm not seeing the peptide in the instruction, so I'm going to deviate from that. Does everyone see this 48180? Are we okay to click that? Oh. So again, here is the accession number, here is the sequence, and I've gone down the page, and I'll do this thing where I close off the sections. So basically by doing this, if I were to go and reload this page, the same tabs would be closed. And so if you're only looking at the peptides or something below that, it's actually quite helpful to be able to go down. So I'm going to click on this accession, this 48180. Do you guys see that? I'm going to select that. And that basically takes you to the peptide interface. So each peptide within the peptide atlas, in the context of a build, gives you this page. And the top one has some characteristics of the peptide itself, some physical characteristics such as the PI, the relative hydrophobicity, the number of samples, and the number of protein samples it was seen in, as well as a proteotypic score, which is an analysis we did for building the SRM Atlas. There are a number of prototypic peptide scores that show which peptides are likely to be more or less amenable to mass spectrometry. This number of builds in which the peptide is found shows that there are actually quite a number of builds that saw this peptide. So I'm going to click on this link that says Compare Proteins. And basically, again, this is going to give us a comparison between two peptides in which this was seen. I'm scrolling over. There's a little scroll bar. So was everybody able to get to this alignment page? So each peptide that has this color, this light blue color, has been observed. And you can see that at the far left, we see the two different accessions. So these are two closely related but distinct yeast proteins. And as we scroll across, you can see that, well, these two have been seen in the same peptide, but this one, or in the same protein, but this one is unique to the upper protein. And as we go across, there are a couple more. This one is distinct to the lower protein. And this one that's highlighted in darker green, or in green, is the one that we came in here with. So basically, we started with a particular peptide that we were looking for, and that's the one that it is. And as you scroll further out, again, these are very highly conserved. proteins where almost always you see a highly conserved amino, or an identical amino acid, and otherwise see, well actually, so the colon is more highly conserved and the dot is moderately conserved and the space is not conserved at all. But these are very similar proteins. So it turns out that for these two proteins, there are two that distinguish each subset. So these probably would have been distinguishable. They would be in separate protein groups, I would hope, in protein profit. So I'm going to click the back button and go back to the peptide information page. So as we said, all these peptides, this particular peptide and all the peptides have been seen a number of times, from one to quite a number. And for each one, we actually have a spectrum. At the top here, there's a link that says show more rows, and actually you can see all of the spectra. If you click that, that opens up this box further, and you can see all of the different spectra for this particular peptide. So I'm just going to click on one of the spectra and hope that it's a decent one. So this is the now familiar lorikeet viewer. We have a very nice contiguous series of Y ions. The B ion series is not as impressive, but there are quite a few. And they're not a whole lot of unexplained peaks. I'm not sure if anyone went over this, but you can actually often explain peaks. So if you look for neutral loss of ammonia, so if you click here, this little button on the side for NH3, you see that there's actually some additional peaks that get identified. And so these are basically the Y4 loss of ammonia, and this is Y5 with loss of ammonia. And so what it shows you is that oftentimes when you have a very dirty spectrum, a lot of little peaks, a lot of them can be identified if you try a little bit harder. But when you're just considering the spectrum and the major peaks, it's somewhat of a distraction, so we don't put those on by default. So all in all, a pretty convincing spectrum. Generally speaking, the spectrum is going to open up in a new window, or a new tab. So I'm going to go back to the list of peptides. And above that, you see that there are a number of, this says consensus spectra. So this, if you mouse over each one, you can see the library that it comes from. And the second one is from the Saccharomyces cerevisiae ion trap NIST library. So I'm going to go ahead and click that one. And it's going to take me back to the peptide, or to the spectrum page, and load that. And you can see that the spectrum that we have now is considerably different. This Y13 dominates. Can anyone hazard a guess as to why this might be different than the spectrum we were looking at? So this is the NIST yeast ion trap library. Do you remember, actually, I was in the wrong build. I was not in the, which type of build? The yeast QQQ build. So the triple quad is a type of instrument, and so the spectra may look different. So if you're working across instruments, oftentimes the spectra will look different. And I'm just going to click the back button to see the previous one. But this is, in fact, a consensus spectrum. One thing that is a little, is both a blessing and a curse, is that, and you can see that actually the B ion series is much more complete in this library than it was in the previous spectrum. The scale on this is always in percentage, so you don't actually see the actual intensities. What you see is a plot that's scaled to 100%, which actually makes it difficult sometimes to judge a spectrum in terms of how intense, how much of that ion was in the mass spectrometer. So again, this is a link where we've clicked on the consensus spectra. I'm going to click back, and basically this tab is going to reload, and this is the previous one. And as we saw, so in the consensus spectrum, this Y13 dominated, and it basically pushed all the other peaks down because it was so high. But here, if you look at 100%, the one that's dictating the scale is actually this Y12. So underneath the individual spectra, basically this plot down here, this little bar chart, shows you which samples this was observed in. And it turns out this particular build doesn't have very many samples. This was a build that was used to make the original yeast SRM atlas. And so we have a number of synthetic peptides, but also this large MRM data set where they did MS2 as part of it. And so we loaded those spectra. But most builds, this will actually be much bigger. And this shows the total number of observations. So in the yeast MRM data set, there were about 85, and in this one, there were about 15. If you instead click on this total obs plot, click on that, this shows the observations per million spectra. And what this says is, oftentimes if you have a data set like the Matthias Munn one that I was showing earlier, that one will dominate in terms of, excuse me, that one will dominate in terms of the plot if you look at the total observations, excuse me. So if you have an experiment that has a lot of spectra, this one will be really big. But if you want to get a sense of the concentration, then you can click on this obs per million spectra, and that essentially normalizes it by the number of spectra that were in the library, so you get a much better idea. So we're going to go back to the search tab. So what we've done so far is we've taken and we've done a search and we've viewed the results, which was a protein. You can also search for a peptide. In that case, you would be taken directly to the peptide page. So we looked at that protein and we looked at the top peptides, and then we selected one and went to look at that. And once we saw that, we looked at some spectra. So there are different sort of objects within the database that are useful to look for. So into the search box, we're going to type MAP1A, which is a gene symbol, and then hit go. Or oftentimes you can just hit return or enter, and it will do the same thing. And you can see we get a number of results in the cow, human, and rat. If you click open the human version, you can see that there are a number of hits. So who knows what this ENSP number is for? Have you guys ever seen this? This is essentially an ensemble protein accession. And then there are a number of, these are Swissprot/Uniprot accessions, where this one is the canonical form or the main form, and this is a isoform. Oftentimes people have the main form with a -1, but as it turns out, the -1 is not always the representative form for that. So we always have the representative form, and then we use their secondary file called varsplik to add in any isoform fields. So we're going to go ahead and click on this P78559. And again, you know that this will take us to a protein page. Oops. So this is a microtubule associated protein and this normal information, and then you can see that it's been seen quite a bit. So it's a pretty abundant protein, and it's been sequenced a lot, and assuming trypsin, it's been sequenced so much, it's so prevalent that we've actually sequenced a number of non-tryptic fragments. So again, if you look at this, we're in the context of the human 2015-03 build. We can also look at it in different ways. So several of you are interested in phosphorylation. So if we go down here and select the human phospho build 2015-09, the most recent one, go ahead and click on that one. And again, you can click go, but it's going to go do this. And in this case, we can scroll down here. We still see almost 270 distinct proteins, 15,000 observations. So even in this phospho build, we see quite a bit. It's not quite as abundant as previously, but notice over here, there's very few peptides in through here. So why do you think that might be? Because there are probably no peptides that are amenable to phosphorylation here, which is actually kind of a nice thing. If you find these areas where there are no peptides, no, when you do phosphoenrichment, you expect to get more phosphopeptides. But there's nothing that says you can't have regular peptides. Always some peptides come along for the ride. But the fact that we have very distinct peptides here and not everywhere indicates that the purification method is actually pretty robust. And it's a long protein, and so if you go down, can you scroll down to this view of the sequence? Do you notice how all the peptides are different lengths here? So that's basically the triptych view. So what we've done is we've taken this peptide and we've broken it up by spaces wherever there would be a triptych digestion. And anyone that's used trypsin knows that if there's a P following a K or R, that is not a triptych site. So those ones are not cut. But you can also click and say interval. And this will give you a more familiar view of the sequence. You can also go and click no space. And this is actually very useful if you want to copy and paste some of the sequence, because oftentimes there are these spaces and you copy and paste it and then you have to go and you have to delete things. But this gives you a contiguous block of text that you can take and copy and paste and do things with. I'm going to go ahead and click this down. So this is the PTM profit view that we showed earlier. I'm not seeing, so this is, so let's pick a protein or peptide that's been seen quite a few times. So let's see. So we're going to examine the peptide SPFEII, which is the top one there. So you guys see it? Yeah, you see it? So let's go ahead and click on it. So I find the phosphorylation a little easier to interpret in the context of a single peptide. So it's taking a little while to load that bit. But again, this is the top section where there's information about the peptide physical characteristics. Here you can see this maps to multiple proteins. And again, if we clicked compare protein, it would show us all those different ones. By default, so in Peptide Atlas, we map to a number of databases. We actually have a hybrid database that includes SwissProt plus Ensemble plus IPI. And what that does for us is it enables us to search by any of those types of accessions. The downside is when you look at something like this, you see quite a number of different entities. So I'm going to click on this, and you can see that this pops up a little window that says alternative mapping. So what I did is I clicked on, when you see the genome mappings, you can see this accession is represented, and then it says 3 more. So I clicked on that, and what it's telling me is, okay, there's this protein. There's this protein that we saw. Then there's actually the main protein we were looking at before and its main isoform, as well as this GI accession that are there. So I'm going to close that and go. Now the page is loaded, and I can tell that because the length of this is, this bar on the side is shorter. So again, we see this localization. So now this peptide has been seen with a single phosphorylation. So how do we know there's been a single phosphorylation? If you're basically if the mass of the precursor ion varies from the mass of the sequence without a phosphorylation by 79 point whatever Daltons, that means there's a single phosphorylation. If it varies by about 160, then that means there were potentially 2 phosphorylations. So you can see again that this represents the confidence that this particular amino acid was seen. And what we see is that in almost, in very few of these 659 observations of this peptide, did the PTM prophet believe that this first serine was the one that was phosphorylated. In about 500, it believed that this was confidently the top one. And in quite a number, it believed that this was the top one. And so these two are essentially indistinguishable. These two are probably both phosphorylated and in part of the population. So these shows the various individual spectra. Oh, not getting a 1. So we can go over and look at one of the spectra. So I'm just going to look at the top one. And this is an Orbitrap spectrum. And it's taken me over to this tab, and it needs to reload it. The sequence is always shown up here. This nomenclature means that, okay, in fact, this one was triply phosphorylated. So this one, the mass of the parent ion varied by 240 from the original. And so in this case, it looks like all 3 of these are phosphorylated. It's a +2 ion. And here is the spectrum. So here we have quite a number of possibles, and there's only 5 of them that are annotated. So this, and there are quite a few that aren't. So this is actually a pretty underwhelming spectrum. We can turn on the various neutral loss ions, and so that's a loss of phosphate. We have a hit, but pretty much this is. So we can increase the mass tolerance and see if that helps. Maybe there was some. So if we increase the mass tolerance, we get a few more hits, but that's almost cheating. So this is not a very inspiring spectrum. So I'm going to go down here and click on. So you see how they're organized by the number of. Basically, they're organized by sequence, and so. These ones down below have an oxidized methionine as well. So I'm going to click on the 3rd spectrum. And this is somewhat more convincing, but again, not a terrific spectrum. So does anyone have any questions about either the peptide or the protein sort of centric view of things? So if you look at these sequences, basically the modification masses are shown in square brackets within the sequence. And so this means that there's a modification on the N-terminus. This 167 means that in this particular case, this serine is phosphorylated. This serine is not phosphorylated. I'm sorry if you can't see this very well. Okay, so this serine has a serine 167. That means that serine is phosphorylated. There's a serine in the middle here, but it does not have a 167 by it. So according to the original identified spectrum, the original peptide spectrum match, this one was not in fact phosphorylated, whereas this serine was. And if you go further, there's a methionine that is oxidized. So M147 is oxidized. The charge is represented here. And I'm going to go back over to the spectrum tab. And within the spectrum, any amino acid that is modified is highlighted. So in this case, all 3 serines are phosphorylated, and the methionine is also modified. And over here it says it's a +2 charge. Oftentimes this has a /2. So sometimes the information is a little bit difficult to see, but essentially in order to see which ones were identified as phosphorylated, you look for this mass modification. Does that make sense? These are from KEGG, and they're essentially metabolic and other pathways. And you can see that there are quite a few here. Right now, the build that we have is this human phosphoproteome. We can change that to say, let's say MTB. We'll change it to MTB, the most recent one we have. And what you'll see is that the pathways change. So this first prefix here, this MTU, stands for the organism Mycobacterium tuberculosis. The human is HSA, Homo sapiens. So those are the KEGG prefixes. So each organism has a code for it. So I'm going to go back to the human phosphoproteome. And again, these pathways will update. And I'm going to click on the glycolysis pathway. So that's just the first one. So if I click on the little PA link, and you also saw this in your TPP output, this essentially takes you to Peptide Atlas when you're outside the resource, or even when you're inside. So that PA is an icon so what you'll see is immediately some information will show up, and I'm going to open that up again. So this talks about what this is. So this is the glycolysis gluconeogenesis pathway from KEGG for human. And it turns out that the pathways are the same for every organism, but for some organisms, some of the boxes are not highlighted. So if we looked at this same one for, say, Bos taurus, cow, or for MTB, we would see the same pathway picture, but different of the boxes would be filled in. The number of peptide, the boxes or the genes that have observed peptides are depicted in green, and the ones that don't have any are depicted in yellow. And the ones for which we had no information are depicted in white. And if you mouse over one of those boxes, you'll see a pop-up. And this one right here, and I don't know if you can see that, maybe not. But if you mouse over these different ones, I didn't mean to do that. If you mouse over these different ones, it will pop up. And the thing that you see is that, can everybody see the little pop-up that says gene IDs and then maybe has list? So what's interesting to me is that each of these boxes actually can represent multiple proteins. And so when you go, you'll see that there are actually multiple accessions in your search box. I'm going to show you one more thing. So as the page loaded, you probably saw some text show up and then it went away. And basically, this is a way you can look at the genes in the pathway. So again, these are entree gene IDs. And basically what this says is looking up genes in the glycolysis gluconeogenesis pathway. And so what we do is we actually go to KEGG and say, hey, I have this pathway. I want to know what genes there are. They send them back. And then we take those and we look in the Peptide Atlas and we find out which ones they are. And then we color the map. Or actually, we get the map and then display the colors. And if you go down here, you can see for all these different things, and again, these are gene IDs, so they don't look like anything. These numbers probably don't mean anything to you. But different of them, this is the number of times they were observed in this particular build. And you can see that a couple of them have been seen an awful lot of times. And so these are probably the ones that are very highly conserved phosphorylations. There are about, this one that has 20,000. That's impressive. So if we go back over here, So this little section exists every time that you run this, but since it's kind of long and it hides the picture, we hide it. And so you can always come back up here and click Hide Stats, and it goes away. And then you can go down here and click on one of the sections. So we're going to click on Enzyme EC. So those are essentially enzyme accessions 513.3, which is this guy right here that represents only a single gene ID. So we'll go ahead and click that and cross our fingers that it's going to go over. And so it's taking us back to the search page. And what we've done is actually for the search page, if you're on an individual page, you already sort of know which accession you're looking for. But the search page actually understands all the accessions. So any accession that we use, you can look for it here. And you can see that we've seen this quite a few times. We typically, more and more, we like to use the SwissProt/UniProt accession. So I'll go ahead and use this one. And I'm going to click on that. And you can see that this one is called canonical, and these are called identical. And the reason for that is that we're using the SwissProt as the canonical. We want each group to be named after the SwissProt protein, even if the other one's identical. And basically, this essentially takes us back to our old view of the protein. So I'm going to quickly go through the menus. So let's say you wanted to get, are there any questions? Are you seeing something that's strange? No, it's okay. So you can download Peptide Atlas builds, and I don't know how to do this in a new window. I'm just going to click on it. So can you do that? Go over to the left and click, and if you can, ask it to open in a new window or new tab, because it's more convenient to be able to go back and forth. And I showed you this as well. This is basically all the different builds that we have. So for any one of these, we can click, or we can download certain data files. And these data files types are listed over on the side. So peptide sequences as a FASTA format. Are you all familiar with FASTA file format? So it's essentially you have an accession that's delimited by an arrow bracket, and then you have, basically you have the arrow symbol, the greater than symbol, followed by an accession, and then a new line, and then your sequence. And so proteins are often in FASTA format, but you can also have peptides. So that's what these are. They have their peptide accession as the identifier, and then they have the sequence. There's a closely related format that I heard mentioned called FASTQ, and FASTQ is basically a DNA sequencing file format where you have the sequence, but you also have quality scores, and generally speaking, they're Fred quality scores. So you can download all the peptides in the atlas as this FASTA format. You can also download the peptides with their chromosomal coordinates. They've all been mapped. You can download the entire build as a MySQL database, or in this case, you can download a spectral library. I'm going to go back. But sometimes you don't want to have all the peptides or all the proteins in a particular build. So underneath queries, there are actually two different ways that you can download a subset of things. So I'm going to click on Browse Proteins. And what this brings up is a form. And I'm just going to go over this very quickly, but this is a common type of form we've seen that I think Ulrika probably showed you. They have these little help things you can do mouse over. If you mouse over the question mark, you get a little pop-up. Or if you click on the question mark, you get this little a little window that pops up, and you can close it, or you can close it by the mark. And basically what this form does is allows you to limit your search. So if I were to just take this, right now my build is human 2015-03, and query, I'm going to get back all the proteins in the build. On the other hand, if I fill out any of these other fields, that's going to filter my search results. And it takes some time to get used to, but it's actually very useful. So in this case, if you had a set of proteins that you're interested in, you can either enter them in freehand here, or if they're in a file, you can upload them in a file, one sequence per line in a text file, and that would basically allow you to download a subset of the build. And you can do that with peptides, and you can do that with proteins as well. You can also compare proteins between two builds. You can select two builds and ask for either the union or the intersection of those two builds. And I'm going to show you one more thing. So I'm going to go down here. So underneath the All Builds tab, go down to Summarize Peptide. And what this does is it shows you the view of this peptide across all different builds. And what you can see is that some builds have far more peptides than others. So by default, you're going to have something here. So how many of you guys actually had this VSFL peptide when you went over here? So sometimes if you've been on a different page looking at a different protein, you get them. Now some peptides are actually kind of interesting in that they're seen in different organisms. So this one is actually seen in Streptococcus, so S. pyogenes, in addition to humans. So that's kind of rare to be in a bacterium as well as human. More frequently, you'll see things that are in mouse and human, things like that. Basically, organisms that are closer on the evolutionary scale. Down below, you see a more familiar plot. Basically, this shows you the total observations. So you can see here that the plasma atlas actually has the most observations of this particular peptide. And I suspect that if I do obs per million, actually now at obs per million, the human is number one. Actually, I take that back. So this is actually, I apologize for this. This is the number of observations per million, and this makes a lot more sense. So what this shows is that this peptide is actually enriched in plasma. But if I click here, it says, now I should show the total observations plot. When I show the total observations plot, you find that you actually saw this a lot more in the human build, or at least more, but because the human plasma build had far fewer samples, it's got a smaller bar. But again, going back to the obs per million, about 600 out of every million spectra in plasma is this particular peptide. And there are also links out to other resources like PASL. So I'm going to go back one more time to the search page. And I'm going to click on one of these example searches. I'm going to click on this ensemble search. So what this is doing, it's a pre-rolled link. So if you've come in here and you don't really have an idea of what you want to look for, this gives you some ideas that you can look for that will return you some reasonable results. You see, since I selected a very specific accession, I'm seeing primarily those. So, and it says over here that it's indistinguishable from this particular protein. So I'm going to go ahead and click in the human kinase build. I'm going to click on this identifier. And it's just been seen 26 times, so not a huge number. So when we get to the protein page, you'll see that it's actually kind of sparse. And again, yeah, you see that various peptides have been seen across it, but not very frequently. So the most abundant peptide has been seen four times. The ones that are mapping to just this protein. Okay, I'm going to go down below. So yeah, so this particular build, I'm going to switch builds to the human all. So the human 2013-04. Oh, no, of course. That's a little. So I'm going to click on the human 2015-03 build. So actually this one was not seen. So in this case, a protein which was seen in a specialized build is not seen in the big build, even though there are far more far more samples in that build. And that shows the utility of having these individual builds, is that they have some things that are specific only to them. So I think that brings us to the end of all the things that we're supposed to talk about. Is there anything anyone has a question about? Again, a resource like this is, it's probably a little hard to understand at first blush. There are a few things that you can pretty quickly find, but it just takes a little bit of playing around with, and hopefully, so if you guys have any, I think you have our email. Actually, the very top of it, there's an email for Eric Deutsch, who I think you should use mine. So my email is dcampbell@systemsbiology.org. And if you ever find yourself wanting, so that's D, and then Campbell is Campbell. at systemsbiology.org. And if you have questions about Peptide Atlas or SRM Atlas or notice something not working right, please feel free to let me know. So we're meant to be over in the main lecture hall soon. We will have a few minutes, but I'm actually going to go down here and show you the Spectral Libraries tab. And there's also a way you can actually use Spectrast to search right on here, but it's only a single spectrum at a time. So at the top of this page, so what I did is I went on the navigation bar on the left-hand side, and if you go further down, there's the Spectral Library links. And I clicked on the upper one, the ones that say Libraries and Info. And this gives information on the various Spectral Library search projects. And as you guys saw yesterday, having the Spectrast search in with your database searches and combining them actually gave you a considerable number of more identifications. So these are libraries that were built from ISB, and actually we have some really big data sets, and we're hoping to basically make this part of each build going forward, in which case these will be quite useful. And these are all the NIST libraries, and you can see that there are quite a few organisms, and we actually keep some of the older ones, which may or may not be useful to you. But you can see that there are probably about over a dozen different organisms. Yeah, exactly. So when you go to the Petunia interface and you ask to add a spectral library, or what is the download spectral libraries, this is essentially the list that you're getting, and then Petunia behind the scenes asks Peptide Atlas, says, hey, we're interested in this particular build, and basically downloads this. And so if you click on the more info, you see that there's some information, including a checksum. Do you guys know why there's often a checksum when you download a file? Does that make sense? So basically, when you download a file, sometimes it's incomplete. And a checksum is a measure of essentially the file's contents, but it's really meaningless except for it is indicative of that particular file. So if you download a file and compare checksums, and the checksums don't agree, then that means that file has been changed somewhat. It may be very simple, a single byte, but it's always good to check, especially if you're downloading software that you're installing. It's definitely good to check the MD5 sum, the checksum. But yeah, basically this is the download link, and this will download all three libraries, for splib and the two indices. And that's what, as Luis pointed out, that's what Petunia is doing behind the scenes.